Well, good morning, everyone. Welcome to our virtual Bench2Market talk. Today, we're going to talk a little bit about milestone-driven research, especially for those who are part of the current Biolocity funding cohort. You guys have your milestone review coming up. So we put together this talk as an opportunity. When we do it on campus, we usually do it more of like a workshop format. So we've adjusted this to a more virtual format, but really to get you guys thinking about how to move from the discovery-based research approach to technology development and into a more product development like state. So we historically, as I said, this has been probably one of our most dynamic sessions that we do. And so we're going to try to maintain that as much as we can in this virtual format. So Brian Walsh will be giving our talk today. I'll introduce him for those of you who don't know him in just a minute. But for all of you on the call, please feel free to jump in with questions, whether that's unmuting yourself. We'll pause at the end on each slide, and if you have questions, feel free to ask them. If you prefer, you can type them into the chat, or Zoom has a raise hand function. You can do that too, and then Christina will call on you. And so with that, look at some other announcements. Go to the next slide, Brian. Maybe. All right, cool. So we have 3 sessions remaining for our Bench2Market talks for this year. Next one is April 22nd. We had initially scheduled physical locations for all of these. We do have virtual backups, and I can tell you that that April 22nd event, License versus Startup, will likely end up being a virtual session. So in that conversation, we'll talk a little bit about how to think about how you exit your technology from the university. Is this best suited for partnering with an existing company through a license or through a spin out or a startup? And we'll talk through some of those nuances, and that tends to be a really dynamic session as well. So I'm looking forward to that. So next slide. Oh, so for those of you who don't know, so Biolocity is a multi-institutional partnership between Georgia Tech and Emory. We provide non-dilutive grant funding as well as business operational expertise. We are also a BARDA Accelerator Network member. And what that means is that we have received funding and support from BARDA to be a conduit for technologies being developed in our ecosystem that address areas of their strategic interest. And as you may guess, right now, nearly all of BARDA's strategic interest is in the area of coronavirus. So BARDA is the kind of the technology subdivision of the Assistant Secretary for Preparedness Response. And they have really expanded their call even in this last week, and we anticipate this to continue to change. So you can look at the BARDA Drive Easy BAA. That is for generally technologies that have an earlier technology readiness level or a full BARDA BAA. And I encourage you guys, Christina's going to put the links in the chat. If you guys are working on things in this area, do take a look at those solicitations and then let us know if there are any that you planned on applying to. We like to track those, and we also, part of our mandate is to help increase the number of on-target and really competitive proposals that BARDA sees. So we don't make the funding decisions, it's still BARDA's funding decisions, but we can help facilitate the process. So let us know if you're interested in any of those. Moving on to business as usual. For those of you who haven't had an opportunity to meet Brian Walsh, he's one of the executives in residence for Biolocity. He has 30+ years of medical device development expertise from big companies like Siemens, so West Coast startups, and then ran North and South America for Dornier, where they made high energy medical devices amongst many other things. And so part of what is really great about Brian is that he's lived that experience of going from unmet medical need to concept to product to post-market. So, you have to live with the thing once you start selling it, and all the fun that comes with that as well. So Brian is the one that we usually tap to give this talk. And again, the goal is to think about your product development in a way that differs from kind of aims-based, more traditional NIH type funding solicitations. And this is something that's helpful not only for your biologically application, but if you're thinking about SBIRs, any of these funding mechanisms, you'll see them moving away from aims-based approaches to go, no-go decision points. And Brian will elaborate on that as well. So with that, Brian, I'll have you take it away.
Great. Can you hear me okay? Yes. Okay. So my name is Brian Walsh, and yeah, I've been on the commercial side for 30 odd years, taking ideas and turning them into real products. So here's what I'm going to talk about today. First of all, generally about planning, which doesn't sound very exciting, but actually ends up being super important. How academic aims and industry milestones are related or not. Value inflection points, because that ties to essentially future funding largely, and also how you communicate with strategics. And then finally, killer experiments, which isn't quite as ominous as it might sound. So planning. So, you know, when I came out of university and I was working in research, I first worked for Siemens, you know, so I was one of 400,000 people worldwide. And it was a little bit like working for the government. But then I got recruited to the Silicon Valley, and that was kind of like the super on steroids education for like how to run a company. And there was one thing, if there's one thing that you remember from this talk, is the definition of a plan. They used to just drill this into us, and we managed a company that grew from 50 million to 450 million in 5 years by doing this every single day. So the definition of a plan is task, dates, and owners. Task, dates, and owners. So I just made this up. It's a very simple example. You see tasks here, and the owner, and then it says start and end dates. So that sounds pretty nominal. As you think about your tasks, so you want to break these into really small pieces. The more granular, the better, and the more control you'll have. As you look about owners, who's going to do it? It's never good to have more than one owner. If you have groups, it's kind of dispersion of responsibility. So nobody owns it. So you really want to focus on who actually owns this. And then start and end dates, and you really want to be very specific. And the one thing I will say is that the units you use as you estimate your schedules matter a lot. So if you're measuring things in days, you know, you might miss by some days. If you're measuring in weeks, you could miss by weeks. If you measure in months. So this resolution becomes very, very important as you do plans. So the other part to consider is cost, and then interdependencies. So some things, you know, you're not going to buy your plants before you select your location, probably, in this simple example. But usually when you start to develop these plans, you can tie it back to your budget. And then what you'll find also is that if you start to come off your plan and you start to stretch out in time, your cost will rise. And it is kind of this altruism that the more time you take, the more it's going to cost you. It doesn't seem like scientifically, analytically right that, you know, you could do the same things for less money, more slowly kind of thing, but it doesn't work out that way. As soon as you start to apply resources, you start to spend money, and you'll end up being more costly. So it's super important to stay on schedule and per your plan. And outside entities like investors and strategic acquirers will expect you to. So this is a Gantt chart. And what this does, it gives you a visual image of kind of like what a schedule looks like. So this is a major task here, and it's broken up into subtasks, okay? And you'll notice these are on days. And also in here, it's hidden, but there's cells that say how much it costs and who's going to do it. Because some of these might be outside resources, like a lab or something. Some of these might be internal resources, etc. But there's a couple other things you can see here. You can actually see how long everything takes in total. You can see dependencies, so you have to finish this before you can start this, right? And you can see critical path, which is going to be down here. So a lot of times when you develop plans like these, and it's important to understand these because both the investors and the strategics live in this world. So you need to become fluent kind of in the space. But the things that you start to see is, you know, at the end, usually when you first cast these, it's too long. It just stretches too long, and you have to come back and say, how do I compress this schedule? And it may be that if you start compressing something in here or up here, critical path becomes down here now, because this is the furthest out. So this whole thing becomes quite dynamic. So the interrelationships on these Gantt charts become really important. So this is what it stands for. I actually had to look this up because I've used Gantt charts for 30 years, but never actually knew what it stood for. It does give you a visual assessment. You can identify the dependencies, as I said, and finding critical path is important. And it gives you a sense for your resource allocation. And you really want to be granular, okay? Really detailed to avoid skipping or missing things. And again, time equals money in these cases. So the longer you go in time, in general, the more money you're going to spend. So it ends up from an academic setting, how you look at research aims and how industry looks at development milestones, they're pretty different. You know, on the academic side, you tend to maximize the understanding of the phenomena. You look at it from multiple directions, right? And the emphasis is on creating new knowledge and kind of complete new knowledge. How does it work? Why does it work? What are the mechanisms? And there is an exercise in, you know, as you all know, in consensus building and a lot of interaction with collaborators, right? And importantly, a lot of the work is done on the front side, and at the end, you document your work. So, you know, this is especially true when you start to see things like software being developed. There's no documentation at all even, which means that, you know, if someone gets hit by a bus, you know, it's all gone. So, but this is the general framework you'll see in the academic setting. Industry is quite different. It's all about time to market and getting things done. So there's lots of go, no-go decision-making. You don't have to find the best way to do it. You just have to find a good way to do it. And there might be multiple good ways to do it, but you need to reach a decision. No decision, when you have options and you reach no decision, you actually are making a decision. And it's not a very good way to manage. So you really want to be active in decision-making. You're focused on how fast you can do things. You want to be capital efficient. That is, you don't want to waste time and waste money. And as you look at risks, you're going to try to address those very early in your process. The more risks you have, the more unknowns you have, the more uncertainty will be in your plan. And this is really important. And this is actually what the FDA says to do as well. You actually document your work before you do it. So if you were to write software or build a device or something like this, you actually write user needs documents, and then you write design specifications on the front side before you do any work. When I first came out of research at UCSD and went to Siemens, you know, I had gone through engineering school, and I'd worked for a team of nuclear physicists writing software in an image processing lab. We didn't write anything, and the first thing the Siemens managers told me to do was go in a room and write specifications for the products they were going to build. And they had, you know, 200 engineers sitting and waiting for me to finish, well, not sitting and waiting, but they couldn't start the work on the new products until I wrote documents, and their documents were vast. There were, you know, dozens and dozens of 3- and 4-inch binders deep. So industry is document before and write everything out before you ever start, which should be woven into your plan. So where do you start? You have an idea. You kind of start to think about the questions that you'll need to address. So what is your technology? And this matters because in a regulated space, the processes are very prescriptive on how you take these things out with medical, right? Do you have IP? Your investors want to know about this, as well as your potential acquirers. Indications and claims, what are you going to say it does? And so the more, as you think about time to market, you tend to adjust your indications and claims. So your indications are things like, I'm going to treat peripheral artery disease with a laser. And my claim is I can do the major vessels. I can do the saphenous vein or something like this, right? Or maybe I can do side branches, and I can do it in under 20 minutes. Those would be the claims for that indication. Who will pay? This is kind of the really big part, which makes medical different. One is regulatory, FDA, et cetera. The other is who will pay. So governments care. Around the world, governments care because they care about the population that, you know, put them in power. And in the US, we have a couple different forms, right? We have Medicare, which is a 65+ population. Medicaid, which is an underprivileged population. We have private insurance, which is not homogeneous. There are hundreds of different insurers throughout the country. And in some cases, like aesthetics, patients will pay directly. So as you look at these, you actually figure out if your technology or your product fits in different segments. So if you're going to have a technology for treating a BPH in the prostate, you're probably going to want to have Medicare type coverage because that's the population that would fit, and probably some private insurance as well, right? And then finally, is it regulated? There's different regulating agencies, as you know. The FDA is the regulating agency in the US. In Europe, they use something called notified bodies. They act as a proxy for the EU in general. They're actually private companies. In Japan, they have the Japanese Health Ministry, which is arguably the most challenging to get your product through. And then, you know, every country in the world has its own regulating agency that you have to apply for typically and get approved individually. So as I said, these are regulated products, and the good news is you don't have to figure out how this process all works because it's written down. It's a prescriptive approach.
Brian, and to interrupt you before you go on this, but you said government care about how you will raise money or how you will get paid for the research or for the product that you're going to develop from this technology?
So the governments don't care so much about paying, but they care about having your product in their market. So for instance, I used to have, when I covered Latin America, you know, I would sell product into Brazil. And some of the terms of their buying agreements were that you couldn't take the product out of the country, or you couldn't leave the country for a decade because you had to support it. So governments care, A, that it's safe and effective, right? But they also care about taking care of their people. And so they have a role in this that is typically more aggressive than if you were selling, say, a car or, I don't know, coffee or something like that. Does that make sense?
Yeah, thank you.
Okay. I'm sorry, I was talking fast. Any other questions on any of the things I went over?
Brian, maybe it might be helpful. We kind of breezed through a little bit of the documentation side of things, but maybe distinguishing the difference between like a lab notebook, which is kind of some of the standard for documentation, versus the generation of specifications and things like that.
Right. So I
just wanted to define documentation a little bit. Yeah.
Right. So the way this normally works in companies, in commercial companies, is they have formal quality systems or formal processes for documentation. And there's typically templates, but it's quite formal. It ends up, your starting place can be your lab notebook, but those early kind of concepts and ideas are then brought forward into formal documents which are under version control, which means if you change them, someone has to say what changed inside, usually on the first or second page, and you know it's initialed, and all those changes are quite formal because the way the process works is you might have user needs, which you write down and are formally codified in a system, and from those, design specifications are created, which also are quite formal. And they might say things, the user needs says, you know, it needs to be smaller than a bread box, and it needs to work within 30 seconds, and it needs to be light and portable. The specifications will say it needs to weigh less than 2.6 pounds. It can be battery operated. So it's quite specific and quantitative. And then when you build your product at the other end, you actually have test plans that you go through something called verification, where you actually test against both your specifications, and then you do validation, which is testing against your user needs. And those are formally documented as well. So you test and prove that the product you built satisfies the requirements for what you're doing or wanting to do in the first place. And all of that is subject to regulating agencies' scrutiny and review. And so when you get an audit, in some cases, depending on your classification of device or drug, you submit all the documentation. In other cases, you certify that you've done all the documentation, you submit part of it, and then the FDA comes out to your site and they spend a week with you auditing you, which is something like an IRS audit. It's not fun. And you know, whatever they find, and then they give you observations or warning letters or whatever based on the quality of your documentation. So that's generally, you know, it's pretty formal, and it is something that's binding and you're expected to have, and it's quite codified by the regulating agencies. Okay. So with that, so I'm going to talk about regulated products. So the world's generally divided into devices and software. It also could be some sort of testing equipment. And then the other world tends to be drugs and biologics, which lives in the therapeutic world, okay? And the FDA is split into separate departments, not along these lines, but actually much more granular than this. But the general process that you go through is very similar, okay? So first of all, you'll have some classification, class 1, 2, and 3. Class 1 is your lowest risk, right? So that's like bedpans. Class 2 would be like a CT scanner. Class 3 is going to be like a permanent cardiac implant, like a heart valve. So they're based on risk. It's important to know whether or not your products have similar devices, or what in a class 2 or class 1 product they call a predicate. So the idea there is that somebody else has already built a product, and the product's on the market, and it got cleared by the FDA. And so you're going to go and behind them and say, we're very similar to this other product, so the level of scrutiny is lower because there's other products like this on the market. And the FDA decides what classification you're in. So for class 1 and 2, predicates become very, very important. There's a paradox here, though. So when to the FDA, you're making the argument that I'm so similar that I'm low risk that there's nothing exciting here to look at, like scrutiny should be low. However, when you go to the market, you're trying to say we're special, we're highly differentiated. So there's a little bit of a push-pull on that particular topic, okay? So anyway, you'll look for predicate devices, and then you'll look for applicable documentation. So for a lot of, for devices, they tend to be FDA guidances often, which are formal documents on the FDA website that say, here's what we want to see, here's what you need to test. There's also ISO or ASTM standards. These are standard, international standards that say how something should be tested or validated. So you'll typically do bench testing of some sort, which, you know, maybe like for a heart valve, you might put it on the bench and have it run in 100 million cycles to show it won't fail. You could, for imaging, you might have phantoms where you have a known phantom and you look at like the contrast and resolution under X-ray, right, between your device and your predicate device. And then if you need to, you'll also do animal work and maybe even human work. It's important, though, for animal work to recognize that you'd like to use animal models that the FDA has already seen before and that people have already used. Otherwise, you're kind of, you actually have to prove the model before you can prove your technology with the model, which makes it much more challenging work and time to market, and it creates a great deal of uncertainty. So having well-accepted animal, and I don't mean accepted from an academic setting, but from an FDA perspective. What do they like to see? What have other people done when they submitted products and had them approved or cleared? And so you really don't want to be novel with the FDA. You want to be very traditional and fit in with what they've done before. On the drug and biologic side, you know, this process is also well characterized, and I think usually you start in vitro, and then you might do small animals. You may move to large animals if there's a model. That isn't always the case. You have to deal with, and usually it comes in kind of in this sequence. You have to deal with formulation, scale. Can you make this in, instead of making it 80 grams at a time, can you make a metric ton? Because if you can't scale it, it kind of doesn't matter because no one can build it, right? And then there's safety, which is a phase 1 trial. They have phase 1, 2, and 3, which is safety, efficacy, and dose. These are separate trials that come in sequence. Usually by the time you get to efficacy, if you're doing a drug or a therapeutic, you're going to exit there, because by the time you get to dose and then to the big human trials that come after this, you're into multiple tens of millions, if not hundreds of millions of dollars. So it's really beyond the scale of a startup. Okay, so usually around efficacy, one of the big pharma strategics would acquire a license. So I want to talk a little bit about value inflection points and risk and cost. So if we look at risk, the red line, you can see at the beginning, you have the most unknowns. All this work's being left to be done. And so the risk is quite high very, very early. And as you do more and more work, risk comes down, right? At the same time, you have no money at the beginning and you haven't spent anything, but the more time and money you spend, the more cost you incur, but that's part of mitigating the risks. Something that's important to note though, if you look at value, value doesn't follow either of these lines. In fact, it isn't a continuous function, it's a step function. So a couple things to note, early on you might have an event and it creates value, okay? And these are called value inflection points. So something happened here that created value. All of a sudden you jumped up and then you did a bunch of work, but your value isn't improving. So say you're doing testing in the lab and you're in the middle of your test, it doesn't, even though you're incurring cost, you don't have your result yet, right? So maybe when you get out here, you get the result of your test. Maybe you come along and then you say, okay, I'm going to write my pre-sub application and then I'm going to submit it to the FDA. And so you have all these different points, but the key thing to note is that these value inflection points are step functions. And the early step functions tend to be smaller in the size of the step. And the later ones are big steps, right? So you're going to do some small animal work. Okay, it gets a little bit better result. Towards the end, though, you're doing things like submitting to the FDA and getting approved by the FDA and maybe doing your first in man, which is a huge value inflection point. So at these points out here, you're starting to drive value up quite a bit. And this is where you'd expect to be acquired typically based on your technology. So here's an example of typical milestones for a class 2 medical device. So this would be like, this could be a laser, this could be an X-ray machine. So it's kind of a moderate risk device. So you might build a functional proof of concept. So most of you now are right here, right now, somewhere in these early phases of building something in your technology. If you're building a device though, and it's also, I'll talk about drugs in a second, but if you're building a device, ultimately you're going to reach a point where you're going to stop. You have to stop working on it. And sometimes it's hard for PIs that are highly vested to say, I can make it better. But the reality is it's better to have something on the market than nothing. So a lot of times, especially with software, you build your first variation of something, knowing straight away you're going to build another generation right behind it, right? There's never version 1.0 software forever. It immediately is followed by 1.1, right? So anyway, you reach a point called design freeze. Design freeze means I'm not going to change the product anymore. So my catheter, I'm not going to make it better or longer or whatever. All the parameters around this thing are frozen, okay? And then I have to go build a bunch of them. And if you're building CT scanners, you might build 2. If you're building catheters, you might build 50. It just depends on the type of verification. That's testing work that you have to do, and validation. So verification, again, tests that you satisfied the design requirements, and validation tests that you satisfy the user needs. So if your catheter, that it has 180-degree flexibility in 2 planes, that the user can hold it in their hand comfortably, and all those types of things. Once you go through all of this verification and validation testing, you have a stack of documentation, and usually it's quite robust. You'll have testing requirements, those documents, and then you'll have test reports, which are the outcome of all these tests. And you'll test statistically large sample sizes. You may need to do sterilization qualification. You may need to do biocomp or other types of things as well. Anyway, all that material is packaged together, and you write an application to the FDA to ask to be cleared. That's called a 510K. So you submit that to the FDA. And for a class 2 device, a 510K, a traditional 510K, the FDA will tell you it takes 90 days to be reviewed and cleared. And the reality is, in my experience, having done dozens and dozens and dozens of these. It seems that you get about to day 85 or day 86 of your 90 days while the FDA is reviewing it. And then they send you a letter, and the letter has a whole bunch of questions. And if the questions are simple questions that you can answer in the next few days, they'll allow, the clock stops when they send you that letter. They'll allow you to answer those questions either in writing or over the phone or both. And then they restart the clock and you continue along your merry way. If though they ask for something like, okay, give us some clinical evidence, and you don't have that clinical evidence, or the question is too complicated to answer in a few days, or the number of questions is so great you can't answer in a few days, you actually stops the clock, and then you submit your answers back and they restart the clock again at like 0. So usually 510Ks take 5 to 6 months to clear. That's just the reality of the real world. At the end of that, you get a clearance. So class 3 products and drugs are approved. Class 1 and 2 products are cleared. But at the end of that, you're actually allowed to market your product. That's what that clearance does. And that's when you make claims that, you know, this is the best thing since, you know, toast. But those claims can't be more aggressive than what your indications and claims you made to the FDA when you got your clearance. So your marketing material and what your salespeople say, et cetera, all are tied back to this FDA clearance or approval. Does that make sense?
Yeah, Brian, sorry,
Yeah.
a question. So when do you submit this 510K clearance document to FDA? This is just after, if it is a class 2 small molecule discovery, just after phase 1, or when do you do this?
Oh, for drugs, I'll do the drugs here in a second. It's a little bit different.
Oh, I see. I see. I'm sorry.
Okay, that's because they have the pathways are different. So
okay.
So here's an example of a development plan. This is a pretty common looking chart, and you'll have charts like this in front of investors and in front of strategics and potential buyers. So on the left here, it has, you know, testing requirements, the usual 510K review, launch product. Manufacturing scale up means you actually start building some volume to build inventory. And you typically do it after your 510K gets cleared, only because if they don't like what you had and you had to alter your product, you certainly wouldn't want to build a bunch and put it on the shelf. CE mark is the equivalent type of regulatory approval for Europe. So you'd submit separately to get approved in Europe, and then you have a European launch. A couple points though, these are in quarters going across. Here's these value inflection points here, okay, called out. And you'll notice that the money is tied to certain value inflection points. So if you're going to an investor, it's kind of you give me money and I'll do a certain thing, right? It's a yin for yang kind of deal. So I need 120K to reach design lock, okay? I need this to get that. I need $1.3 million to become cash flow positive, right? I need 350K to get through whatever the annualized sales are greater than $10 million, right? That's the form of this. And so when you approach investors, they'll expect you to have an ask, and that's what you're doing. You're asking, but when you have an ask, you don't just ask for money. You ask for money to achieve a goal, and the goal has to be something they value. So FDA clearance may not be enough that they value it, right? You actually may have to be able to sell the product and demonstrate that people will pay for your product. That's what this cash flow positive does, right? And they're like, oh, there really is a market for this. Other times, it may be that all you need to do is get cleared or get CE mark. But in this case, you needed to get to a point that you prove the market out a little bit, okay? The other thing you'll see is, you know, the value in the middle of this isn't great, right? There's no milestone. It's always at the end of something happening significant, okay? And it's also true for drugs. I'll show you an example in a second. So this would be a drug type example. You're going to have a proof of concept probably of lots of drugs here. You'll say, I have, you know, 30 molecules. Well, the problem with having 30 molecules is all of this work is really hard to do and really expensive and super time consuming. So you're not going to work on 30. You're going to kind of refine and say, okay, here's my best bet. And they call that the lead, okay? So we have a lead here, a lead candidate. This is your best molecule, your best drug, or your best biologic, whatever it is, okay? And this is kind of the equivalent of the design freeze from the device, okay? And then you're gonna actually have your candidate, you're going to do a whole bunch of testing. And I'll show you some of that testing in a second. And then you file for an IND. That's an investigational new drug application. And then you start to various clinical trials. So this is phase 1. So this is going to be safety, efficacy, and dose, right? As you go through these, usually around here, you're acquired, okay, by somebody. You actually don't take it all the way through all this stuff. This application for the IND just allows you to do the testing in humans, okay? Before this, though, you did all the small animal work, the large animal work on the lead candidate. You're going to do, you know, tox and all kinds of different stuff that the FDA requires in order to, and submit it with your IND to say it's both safe and effective. The FDA always cares about safety and efficacy, with the emphasis typically being on the former. So you as an academic researcher will probably never actually submit to the FDA. You're going to get, you might get through this for the application to start testing, but somewhere during the middle of your testing, someone's going to acquire your technology because it's just too expensive and too hard to get through all these trials and actually get approved. It's just, it gets into the hundreds of millions of dollars, okay? So this would be, for drug development value inflection points, the same chart. So you kind of end up in this preclinical work here at the inside the university. So this could be your, you know, doing something on bench. It could be done with small animals, and then maybe if it applies, you go to large animals. When you start to do the clinical development, right? Then you end up doing this phase 1 stuff, maybe phase 2. You'd be a biotech company, right? So you might have a small startup and doing that type of work. But somewhere in this process, large pharmaceuticals acquire and finish it. Okay, so your approval ends up being with the large pharmaceutical companies who are well suited to do this and know actually how to both get the approvals and commercialize the product. Okay, so again, you do have value inflection points when you finish certain things. But there are different value inflection points because it's a different pathway and you have to run different types of tests. Okay. So this would be kind of what a Gantt might look like. This was for a drug. So, you know, here's the formulation optimization. So that's a major milestone that's broken down into a series of smaller tests. And all of this stuff is very prescriptive. They're either guidance documents or the other thing, and we help you with this. If you have something that's quite special, unique, we look for regulatory consultants who have done this before in the space. There are a lot out there that are former FDA. But I will tell you this, when you're securing regulatory guidance on what testing, for instance, and how to map all this stuff out, you know, and who's going to do it, you're really paying for, with these regulatory people, their past history and past experience. There isn't enough written down to that they all. You really are looking for them having been in your specific space before and knowing all the perils of getting through that. And I'll give you an example. I'll give you a device example because I worked more on the device side. So we built, one of the products we built was medical lasers. And to the FDA, they're all class 2 devices. And they don't care what the wavelength is, but they do care about the indication. And so there's like these unwritten rules. When I submitted lasers for approval for vein closure, I just wrote up what it did. I said, it's just like these other guys that have vein lasers, and submitted, and I got approved in however many months. If you were to do the same thing for prostates, they would require you to have several hundred patients in a clinical trial and submit the clinical trial data. So in one case, even though they view them in the same classification, they're similar devices, they don't even differentiate between, and the two types of lasers would be different lasers, different wavelengths and different energy levels. But in one case, they wanted a clinical trial, in the other case, not, and there's no way of knowing that unless you worked in the space before or found other other products and found their submissions to the FDA and whatever they went through in terms of addressing it. So the rules are kind of moving rules a little bit, which is why you need help to figure out what all these tasks are and then figure out who can do all those. And you'll end up, some of these, in this case, this is an Emory project. Some of this can be done inside of Emory. Some of these will be done like with an outside clinical research organization. And some of the stuff you can't do in the university because you're not certified or you don't have formal processes set up in a way that would be accepted by regulatory agencies. So there's a bunch of stuff that has to be done by outside resources, especially with drug development. Okay. So typical value inflection points, and not all these are going to apply to all projects. You kind of have to pick and choose which ones, make sense for you. Having patents filed or issues a big one because that actually says you have something. Copyright would be another one if you did software, for example. Completion of animal work with results, or a proof of concept of a technology that having a lead or having a proof of concept model of your technology, reaching design freeze or finishing your verification and or validation testing, having done your first in man is typically a large value inflection point that strategics really respect. Getting a 510K filed or cleared, having an IDE or an IND pre-meeting with the FDA, those are all good value inflection points. And investors typically ask questions like, are you in discussion with the FDA? And the answer is yes when you're doing all these types of activities, because they want the FDA to weigh in on your plan because a lot of times they change them. And so it's de-risked more if the FDA has seen it and is in agreement or you've adopted to whatever the FDA has recommended. And then first commercial sales is another good one. So let me stop there. Questions?
Yeah, I have a question about your last slide, and you had said completion of animal work as one of the inflection points. But I feel like animal work could continue a long time because there's always different questions you could ask about, I don't know, whatever you're trying to develop. So can you explain that?
So let me say it this way. If you're trying to get a product to market, you're actually looking for what is the minimum amount of work you can do to satisfy whomever. It could be investors, it could be in strategics, or it could be the regulating agencies, right? So what you say is true. Matter of fact, I'm going through that right now on a project I'm working with. We've completed chronic animal work in large animals, dozens of them. And the PI has decided, he's like, oh, we can start another set and we'll study this question. And I'm great, but don't tell the investors that or the strategics because then they'll say, okay, great, I'll wait for those results. So this is kind of a case of you're looking for your minimum requirements, and you have to figure out what your stopping conditions are. And sometimes it's hard to stop. But the general rules are going to be around the structures of safety and efficacy. And if you're in small animals, and you see this a lot with drugs, right? You could go from mice actually, and that'd be enough. In others, you actually have to go to large animal models. But it really comes down to figuring out what's the minimum requirements, if that makes sense, as opposed to what's the interesting question, because you can do that forever, and you still can do that work. You just don't make it part of your story and your product development, right? It's kind of outside that scope, because you want to have a discrete plan, and you want to execute against that plan. And so the more open questions you have, actually the more risk you have. The more risk you have, the more you can't control cost and time, right? And so people don't like that so much. So you're trying to by doing the animal work, you're trying to put certain things to bed, right? And you're de-risking it, but you want to not introduce additional work that's kind of beyond the scope of what is minimally required to bring a product to market. Does that make sense?
Yes, thank you. That's great. That's a great explanation.
So basically, Brian, on the design freeze, basically by the time you locked in your molecule, whatever it is, you locked in your disease, and you just, that is the one, that is the route, that is the molecule, that is the disease, you're going to go in for first in human. Is that
Well, you're going to do all this stuff that's on the left first. This is all the testing of all of this stuff.
Yeah.
Right? You have to do all this stuff, and they're doing it in rats, I guess. But you know, you do dose escalation, a bunch of other stuff in animals first. And once you have all that stuff, then you have to figure out this part here is like manufacturing. You have to figure out, okay, I've done all this stuff. Can I scale it? And then I have to prove it. And then I can ask for this pre-IND meeting. And that's the part that gets you into first in humans.
So said differently, the lead candidate milestone is where you say we have enough data here that we think we can take the big expensive bet on doing the tox and doing all this GLP work. You know, it's there is sufficient evidence that you're going to move this one forward and put substantial resources to do the massive battery of testing you need to do to get to an IND.
Okay.
Okay.
So, okay, I'm going to shift topics here to killer experiment. Okay.
Brian, we have about 10 minutes remaining.
Okay. I will talk fast. So, a killer experiment essentially involves the idea that you want to figure out what your greatest risks are right now and either mitigate those, put those to bed. So, if you have to do a test, you have to do something to mitigate them, great. And if not, and they fail and you can't mitigate them, you want to kill the project or pivot the project because you don't want to waste your time. That way you have the chance to move on to something that, you know, is more promising. So these are the types of things that the FDA looks at, the funders, the acquirers, they all look at what are the risks, because, you know, risk equals uncertainty on time and money, okay? So I'll give you an example real quick. This is a transcatheter delivered heart valve, and there's a working channel in the catheter, and the largest catheter that you're allowed to use is 16 French. And so the killer experiment is, can I create a valve that fits inside a 16 French working catheter working channel? Okay, and so they actually had to go and do this and then perform surgery in pigs. And that kind of proved it out, because before that, you know, they couldn't fit inside 16 French. Another example was we had a team that was developing a low-cost glass ceramic material to replace crystals using X-ray equipment. There's, you know, X-rays are invisible, so the way X-ray works, X-ray hits these crystals, it gives off light, and the light creates an image. The crystals are really expensive. Like a nuclear medicine gamma camera crystal could be $30,000 to $50,000 just for the crystal. And these guys had a glass ceramic process to make scintillators like this, and you know, it cost hundreds of dollars. So it was, you know, an amazing technology. What they had to do though is that they started to run phantom image tests against the current crystal technology, and it found that the performance wasn't as good, and therefore they couldn't sell it. So this team stopped working on a medical application and pivoted towards a security application. Like, I guess they would use these for scanning trucks with radiation inside or something. So they, you know, killed their project. The one above mitigated their project, or mitigated the risk. So I'm not going to walk through, I think, all of this. And again, this is the development plan. Okay, so this is the last slide. So as you think about what to do next, you want to determine the tasks that need to be performed and do it at a high level of granularity, very, very detailed, right? Then you identify the resources who can do this, and you'll end up having both time and cost for each of this. You'll look for the dependencies. What do you have to do first, and what do you have to know the outcome of one thing to do the next? And as you do your planning, there's this natural tendency to buffer things and say, okay, I'll add 30% to this, give or take. You really want to be able to keep breaking it down in smaller and smaller pieces so that you can actually prove that, in fact, it is 8 days. It isn't 2.5 weeks-ish or something like that. You actually want to figure that out at a very, very detailed level, and then you can estimate cost. And you need all of that because your acquirers, as well as your investors, will expect all of that type of material, okay? And when you go through due diligence with your investors, they're going to look at your budgets, your plans, and based on their experience, you know, these make sense or they don't. Yeah. Questions? that's all I've got. Okay.
I have a question, but I'm still sort of formulating it. Some of your milestones are things like product development, like for a drug, can you mass produce it? Or for a device, can you mass produce it? Should we be thinking all the way there, or is that something that sort of comes later in the process that we're not going to?
You'll have, yeah, if you're doing a drug, for a device, whether you can mass produce is typically less risky. It's usually more for drugs, unless the device has some like super expensive component that makes it crazy expensive and outside the market expectation. So for drugs, you will have to address, can it be scaled? And someone will help you with that. There's outside groups that do it, and you'll have to bond with those outside entities. But it becomes part of your problem as you go through this, because it's great if you can make 5 grams, great. But if you need to be able to sell it, right, and the process is so complicated that they can't manufacture it in volume, that's a problem.
Yeah, and on the device side at this early stage, it really ends up being kind of just a sanity check. So if you need let's say it's like a rare earth metal, and to get to scale for your market, you would need 3 times as much as is currently mined in the world. Like that's a risk that you would need to address sooner than later. But it's those kinds of sanity check for scale ups, that's kind of all that's necessary right now. So if there's no obvious immediate risk, then you're generally okay.
Other questions? Okay. Well, thank you all for your attention and your time.
I have another one. So one of the things that's unclear, I think, at this stage would be, when do you need to go for full trials, like phase 1, 2, and 3? And I think you said if you have a similar, for devices, if you have something really similar, you could say you don't need phase 1, 2, or 3. But you don't really know until you talk to the FDA. Is that how that ultimately gets decided?
So for drugs and biologics, you have to always do it. For devices, a class 3 device, and the FDA decides your classification. If they haven't, then you have a meeting with them and pitch your case, and they tell you what they think. If it's a class 3 device, you do have to do a clinical trial, typically, and so you'll end up doing, the ones I've done were multi-site, multi-million-dollar over multiple years. If you're class 2 or class 1, most class 2 don't require clinical trials. However, that said, if you were building a piece of X-ray equipment, there is a guidance for imaging that says you have to run imaging experiments on the predicate device and your device, including patients, and then have radiologists read them and give them a score, and you submit the score to the FDA. So there is some limited work, but typically the amount of clinical work is pretty low for a class 2 if it's required.
So,
sorry, I have a question.
Sure.
Can you hear me? Yes?
Yes, yes.
So we are developing a software, and because when you develop a device, sometimes they come with a software, but in our case, it's more like an app. So in that case, is it a medical device or is not? And how much testing do we need to do? Because it's basically to facilitate the selection of different drugs for chemotherapy. So we need to show that we can predict the effect of the different drugs in different patients. But we don't know if we need to do for 100 patients or for 1,000 patients, because it doesn't really require a clinical trial. So I don't know if you have any advice.
Well, apps are a moving target right now. There were some guidances that actually came out recently from the FDA, but let me just, whether it's an app or not, and you'll want to probably have a pre-sub with the FDA, which is a pre-meeting to get some guidance from them. But here's kind of how I think about software. if you have software that provides information, that's great. If the software tries to replace doctors' decision-making, okay, then you get into a very dangerous place with the FDA. So, you know, and I'll give you an example. I've seen software for cardiac perfusion measurements, and it comes out with, you know, colored images that indicate percent stenosis or whatever, right? That's all fine. But if that software said this patient needs a stent on its LAD, that would be a problem probably for the FDA, because the software is trying to do what a doctor might. So the way you do this normally is you provide information to physicians, and physicians make a choice. Okay, so the physician's always
that
in the
will
loop,
be the case. Yeah. Mm-hmm.
and it needs to be in that form for them.
And on terms of testing.
So in terms of testing, it's going to depend a little bit on what you claim it does, because you know you're going to define user needs, and then you're going to write specifications around it. Those design specifications, the amount of testing you have to do, that's the kind of software or the kind of testing, the verification testing, that, you know, it doesn't crash, or it doesn't come up with negative numbers, or it doesn't miscalculate something. That's the kind of testing you do there. So you're going to have test plans and then test reports and somebody who runs those tests, which is going to be different than the developer, because you can't be the developer and the tester because it's a conflict of interest, right? And then the other testing you need to do is validation testing, where you test whether or not it actually satisfies the user needs. But I think in your case, it's going to matter really a lot about what you claim it does. So there's certain things that, you know, if you're in the industry for a while, you start to, you know, to be careful around. And I'll give you an example. At one point, we made a urological table, and we started to make safety claims. We said it's safer than the other type of table because of this. If you make a safety claim like that, you actually have to run a safety trial, okay? If I didn't claim it was safer, I just said, you know, it's a really good table, and it goes up and down, and it moves this way, and I didn't say it was safer, I don't have to run that trial. So it's going to matter a lot about what you claim it does.
Okay, thank you.